Hi everyone, welcome back to BMT 311 Immunology, and we're now continuing into the next lecture, which is immune response to infection. So last lecture we talked about immunological memory and vaccination, and now we're going to go into the different types of infections depending on the types of agents that we have. So by the end of this lecture, I hope you'll be able to describe and differentiate the main features of the immune response against viruses, bacteria, parasites, and fungi. And I mean, obviously there's so many viruses, so many bacteria, so many parasites, so many fungi. But I decided to choose basically some models for us to discuss the differences in the immune response. Some of these are biased based on my personal interest, but they're also some of the key ones that are important from a global public health perspective as well. So we're going to talk about influenza. Obviously, because now of the coronavirus, we'll also touch a bit about the coronavirus, but more in detail about influenza. We'll talk about tuberculosis for a bacterial infection, and we'll talk about Plasmodium and Schistosoma for parasites. And then we'll touch a little bit about some opportunistic fungal infections and how the immune response differs against these. Right, so you've seen this before, and again, this is a very oversimplified version of what attacks what in a human body. So I'm not going to go too much detail. So before we go into specific types of infections, let's just recall the immune response in general again. So it usually occurs in three phases, right? The first is the physical barrier, which should stop entry in the first place. As long as something cannot enter you, more often than not, it cannot make you sick, okay? There can be some superficial types of infections, but those don't usually make you very sick. They're usually annoyances. The physical barriers like your skin and other mucosal tissues that are lining, for example, your intestines or your throat, they're all secreting things that should prevent adhesion and penetration, or they have conditions so they secrete things that change the pH of the environment, or they have secretory antibodies that neutralize the pathogen before it can even enter. So those are your physical barriers. And then you have the innate immunity, right? So we remember that the innate immunity is sort of like a general immune response. So the foot soldiers that just kind of handle the first level of the battle. So they recognize pathogen-associated molecular patterns, or PAMPs. And they're basically macrophages and neutrophils that will usually engulf, phagocytose the foreign organisms and then kill them. So that's their mechanism. They'll just eat it up, and then they'll kill them. You'll have some plasma proteins called the complement system. So they also target the pathogens to lyse them and also enable the macrophages to gobble them up. But then we have our more sophisticated branch of immunity called the adaptive immunity. So this kicks in after the innate immunity realizes, okay, I can't get rid of this fast enough. And at the same time, let's just call for backup here. So they'll stimulate the B cells and T cells to undergo clonal expansion, and then they will also get involved in trying to clear the infection and the pathogen through antibodies or through cell-mediated responses. So the first type of infections we're going to talk about are viral infections. So just as a recap, so I'm sure you remember viruses are these creations, or structures. We don't know if they're alive or they're not alive, but basically what they are small segments of nucleic acid or their genetic material that has a protein or lipoprotein coat, and they are what we call obligate intracellular parasites. They do need to be in a living cell in order for them to make more copies of themselves. Otherwise, they cannot grow or replicate. And so Sir Peter Medawar, Nobel laureate, calls viruses bad news wrapped in protein. So they're so very tiny, much tinier than bacteria, bigger than prions. We're not going to talk about prions here, but prions are even scarier because they're like just proteins that can really make you sick. So at least this one has genetic material, and it makes sense that they can make multiple copies and make you sick. But there are also different kinds of them, different shapes. So you have, and also different types of viruses that infect different cells, different types of organisms. So bacteriophage will infect bacteria. So they are our friends often. Then you have the common pathogenic viruses like influenza, adenovirus, HIV, hepatitis B, Ebola, and of course our very familiar SARS coronavirus 2. So the virus infections start first with entry. So the virus needs to be able to have a receptor that it can bind, and often this is like an enzyme of some sort, or some receptor that's found on the cell that has some other purpose, obviously, but for some reason the virus also is able to bind it. It recognizes the viral antigen, and then it unlocks and allows the virus to enter. So that's when, once the virus enters, that's it. What it will do is it will start using the cell to replicate itself, to the point that it destroys the cells. So the virus is more likely to survive or thrive if it doesn't actually kill the host. So it kills the cells, but if it can kind of have this level of replication where it doesn't rapidly cause the host to die, it's actually much happier, because then it can also be spread, and then it can sustain generations of viruses. It's difficult to, because they're not really living, but yeah, they don't have offspring, but they have multiple copies of themselves, right? So they actually want the host to survive, so that the host can spread the virus to others. But sometimes, and this is what happens to certain viruses that mutate a lot, such as the coronavirus, the mutations can sometimes produce lethal mutants. So when they produce lethal mutants, it actually often causes early death of the host. So that's not ideal for a virus that wants to spread itself as much as possible. So what does it do? In order to survive, the virus then needs to be able to spread to new hosts as rapidly as possible. So it kind of sounds familiar now with what's happening with the COVID-19, right? So the COVID-19 is caused by the SARS coronavirus 2. So the SARS coronavirus 2, like SARS, is characterized by these spike proteins on top of its envelope. So the spike is what is able to bind to the ACE2 receptor, or the angiotensin converter enzyme, which is found on a lot of cells, but mainly, there's a lot of them on our lung cells, right? Our type 2, our alveolar cells. So because the spike 2 recognizes these ACE2, it's able to bind, it's able to enter the lung cells, and then it gives us all those respiratory symptoms, like coughing and shortness of breath if it's quite severe. Now, what's interesting is, compared to SARS, SARS is not as infectious. It's actually more deadly. Because it causes more people to die, more infected people to die, compared to the SARS coronavirus 2 so far. But the SARS coronavirus 2 is able to bind the ACE2 much higher affinity, like 10 times higher than the SARS, which means it's able to be spread much faster because it's so transmissible. Yeah, you don't need a lot of it to start being able to enter the cells and then replicate, because it's able to bind so efficiently to the receptor. So what is the response, once we know that there are some of these viruses that are infecting us? So the immune response is both humoral and cell-mediated, but the most common cytokine produced in response to viruses are interferons. So they're able to inhibit the viral replication by inducing an antiviral response in the neighboring cells. So the viruses are also controlled by natural killer cells. So they're the first line of innate response against viruses. So this table, you can go through this later, but this kind of tells you what the effector molecules are and what they do in response to viruses. And what I want to talk about for the flu specifically, because the flu is very interesting. The flu is a viral infection that will never go away. Even though we have a vaccine, obviously the vaccine cannot keep up with the rate of mutation that the influenza virus undergoes. There's also a lot of strains of influenza. So time and time again, there'll be waves and epidemics or even pandemics of flu. And the last pandemic we had was in 2009, the swine flu. I don't know how old you were then. I was 10 years younger. And it was quite terrible. Of course, not as terrible as this COVID-19, because at least flu, we had a vaccine. And flu is a bit more familiar. So this coronavirus is a new thing that causing a lot of uncertainty. But what is it about the flu virus that allows it to mutate and, in a sense, evade our immune responses? Because it seems like a new virus each time. So it's not so recognizable because it changes so fast. So what drives it to change is its mutation rate. And what it does is it creates antigenic variation between flu strains. So there are two main mechanisms that produces this antigenic variation. The first is the antigenic drift, and the second is the antigenic shift. So with the drift, it's basically a series of spontaneous point mutations that occur over time. So it's actually minor changes in the antigens. The key antigens for the flu are the hemagglutinin and neuraminidase. So these you can see here, hemagglutinin and neuraminidase. So they'll change their structure very subtly over time, but it's still changing. Then you have antigenic shift, and this is when you suddenly have a new subtype of influenza. So the HA and the NA are very different from whatever virus that was present before. So this is basically an issue of time. Does it change very rapidly, or does it change gradually over time? So that's the drift and the shift, yeah? And another thing that we talk about for the immunity against these changing viruses is the idea of the original antigenic sin. So we know that you'll have preformed immunity from primary exposure or primary response. So that's the case that we have for vaccination and also for natural infection. Whatever infection that you've gone through, whatever vaccination that stimulated your immune response will give you some protection, sometimes lifelong protection from pathogens that have stable antigenic presentation. But it also neglects subtle changes in pathogens that mutate and change. So like the flu virus that we were talking about just now, some of it, they'll change slowly over time, yeah? So the second time we meet a pathogen that has many similar antigens that was seen in the first encounter, it'll only trigger the memory cells. Okay, so the primary response to new epitopes that the pathogens carried will not be made. It purposely ignores anything that looks a bit different. So it's just, okay, oh, I recognize this. I've met you before. Okay, memory cells, come on. Let's just produce that secondary response. But you don't mount a primary response to any of the antigens on that same pathogen that is different. So you selectively ignore and you prevent a primary response from being produced. Okay? And this is because the Fc of the antibodies are binding to the FCR of the naive B cells. So that's kind of in a way how it's putting blinders on the naive B cells from mounting a primary response. What happens is that over time, yeah, as you keep on, let's say you keep encountering these subtle changed flu viruses. So the first time let's say you get vaccinated or you get the first flu infection, and then you now have memory against that particular flu strain or subtype, and then you get infected again, right? But you don't get as sick. Maybe you don't even get sick at all, because the memory cells have now recognized as a majority of the antigens on that the other virus. But the virus is not exactly the same though, yeah? And then next time you meet another one, because the virus is also mutating, so the circulation, it's circulation here means it's going around. It's being spread from one person to another over time, and by the time it comes back to you, it's changed, it's mutated already. And so by the time it comes back to you the second time, you're ignoring anything that's different about it, only mounting a secondary response to what was familiar. And then the third time comes, okay? The third time, let's say, here, The third time, you'll still be able to use your memory response. You only focus on what you recognize, which is, for example, this particular A epitope. Ignore that the E and D is now there and you don't recognize it. But then the fourth time, by the time it comes back a fourth time, it's mutated so much, or it seems like it's mutated so much, that there are no more antigens that your memory cells recognize. So now you will mount a very big primary response. Yeah? And that's usually what gives you a bad case of flu, that fourth time. Because previously, you were ignoring things that were changing over time very slowly. And now when it comes, you're very shocked that, oh, you're so different now. and then, yeah. But basically how it works. The original antigenic sin. Another interesting thing about viral infections is the influence of aging on it. So if you notice certain viruses, I mean, this happens with a lot of infectious diseases. Usually it will affect the youngest and the oldest. And this is certainly the case for flu. You have a lot of deaths that occur in people over 64. But what's interesting is, for the COVID-19, you see this severity in older people, but you don't see that in younger people. So we're not sure why exactly. But there are some possible reasons why these viral diseases are worse in elderly. It's usually because they also have other chronic illnesses. And for COVID-19, that's certainly a factor as well. When you have comorbidities, it doesn't matter how old or young you are more likely to get a severe disease, a more severe outcome from the infection. So yeah, comorbidities, or generally you have a weaker respiratory system, and also because just being older usually it impacts your innate and adaptive immune system. I'm not going to go into a lot of detail of this, you can read it, but in general you have certain impacts on innate immunity and adaptive immunity as you age. And there's some data about this. It's kind of like a developing area, but what people have observed is, for example, for T-cell immunity in flu infection, your CD8 T cells are impaired. You don't have as many CD8 T cell viral reactive interferon gamma responses as you grow older. And so this also may be a reason why when you try to vaccinate older people, they actually don't mount a good enough response. Now, just to switch back to COVID-19, what's interesting is, again, we know that it's more severe in people who are older. We know it's more severe in people who have more comorbidities. And we know that aging usually decreases your immune system. So if aging impairs the immunity, why is it in COVID-19 that severity or the fatality that comes from inflammation occurs? So let me repeat that. The virus makes you sick, but what kills you often is when you have too much inflammation. And where do you get inflammation? You get it from your own immune system. Your immune system is producing destructive responses that destroy your own cells. So why does that happen in COVID-19? So that's a question that people are trying to answer. Something to think about. All right. So now we're going to switch gears a bit. So just now we talked about viruses. Now we're talking about bacterial infections. So with bacterial infections, obviously, they are not obligate intracellular parasites. They can replicate on their own extracellularly, but some of them also can go intracellularly. So it depends on whether these bacteria that are infecting a person is extracellular or intracellular. Another factor that plays into how the immune system responds is the inocula, which is how much is actually entering, how much of the bacteria is actually entering, and then the virulence of the bacteria. So if it's got low inocula and low virulence, usually just the innate response or the local phagocytes can get rid of it. But if you've got a high inocula and a high virulence, then usually the adaptive immune response is induced to get rid of it. And when we talk about that, adaptive immune response, it also depends on intracellular versus extracellular. If it's extracellular, it's usually generated or mediated by antibodies, which then activates the complement system. The antibodies also neutralize the toxins, and the antibodies serve as opsonins to increase phagocytosis. But if it's intracellular, it largely depends on CD4 T cell mediated responses and delayed type hypersensitivity. Right, so yeah, I'm not going to go into this in detail. You can have a look at it, but this is basically describing how the antibodies work in combating an extracellular bacterial infection. So these are, again, some more details about the host defenses and how the bacteria evades these mechanisms and the processes for the infection. Okay, now we're going to talk about our model bacterial infection, which is tuberculosis. So tuberculosis is an intracellular infection. It's a bit different from extracellular infections, in which it actually enters the cells and then replicates within the cells instead of outside. So tuberculosis is caused by Mycobacterium tuberculosis, and it causes respiratory infection, usually deep in the lung, and it starts when you inhale small droplets of respiratory secretions that have the bacteria. So the bacteria are first ingested by alveolar macrophages in the lungs, and they're able to survive. So they're ingested, but then they'll actually multiply inside of these alveolar macrophages. And how they do this is when the bacteria is phagocytosed, usually it will then fuse with lysosomes, so that the lysosomes can produce all the lysozymes that then break up the cell. But what the bacteria does is it actually prevents the fusion of the lysosome to the phagosome. So this phagolysosome is not made, and so instead of being destroyed within the cells, it's now in like a nice little pocket that it can use to multiply in. So when the macrophage has a lot of bacteria growing inside it, obviously eventually it will die, and it will also be lysed, and then it'll release a lot of the new bacteria that has been produced inside it. So this is plan A usually. Plan A is to, if this was the macrophage, is to eat up the bacteria and kill it, but it obviously fails because instead of killing it, you get more copies, you get more bacteria, and then what happens is you have a lot of then dead macrophages with carrying the TB bacteria. It's a lot of words, bear with me. So because we've got basically that failed plan A, which is to use our macrophages to phagocytose and kill the bacteria, now there has to be a plan B. And this is where the feature of TB is very distinct. So once you've got a lot of these macrophages that have been activated and contain the bacilli, the CD4 T cells get activated, and they start secreting cytokines to call in more activated macrophages, and they'll start clustering around the infected macrophages. Okay, so you've got activated macrophages surrounding the infected macrophages, and then they'll start forming sort of this barrier, or what we call a granuloma or tubercle. So the cytokines that the CD4 T cells produce are really important in maintaining the structure. So even though they couldn't get rid of the bacteria, they've now been able to cut off the bacteria from spreading because it's now trapped inside the granuloma. And in a lot of people, this granuloma can be containing the bacteria well until they die, and they never know that they had been infected with TB because they never felt sick from it. But in some people, about 10%, the granuloma actually does not resolve. And then because there's mycobacterial antigens accumulating, the CD4 T cell activation somehow will start turning against the structure of the granuloma, and it will actually lead to a high concentration of lytic enzymes inside the granuloma. So this lytic enzyme causes lysis of all the cells, and it starts turning all the cells in the middle, a bit like a soup, like a medium. So you know how we use the medium in the lab to grow the bacteria? Now you have medium within these granulomas that allow the bacilli to grow even happily. Apologies. So it's growing extracellularly. I'm going to pause. Alright, so let's resume. I got my water. Just basically doing lectures back to back. Okay, where were we? Yes, so now that the tables have turned, so the granuloma that was supposed to contain these bacteria are now sort of like these little mini vessels of media, because the cells that contain the macrophages that were infected with the bacteria that were kind of containing them before have now lysed, and they've now become like a really rich medium that allow the bacteria to suddenly grow very rapidly. And if you recall, originally the bacteria were only replicating intracellularly, but now they're outside of the cells, but they're replicating as well. Now this eventually causes the tubercle to rupture. So that these little balls that were trying to contain the bacteria break apart. The bacteria then escape, spread in the lungs. This is the most common form, which we call pulmonary TB. The person starts coughing, spreading it out to people. Or sometimes it also enters the blood vessels and then it travels elsewhere. And then it starts creating new colonies of bacteria in these other parts of the body. And we call that extrapulmonary TB. Yeah, so this basically is a little funny cartoon that I made to show what happens. So if you've got it contained, you call it intact granuloma, and then if basically this has turned into soup, then the bacteria starts multiplying, it ruptures, and then it gets out. I think this can be made nicer. I wish I had better skills in drawing cartoons. This is another schematic just to show it again. The granuloma that is sort of resolved and the ones that aren't resolved, and kind of break apart and release the bacteria, causing what we call an active infection. So before that it was a latent infection when you have the granulomas intact. All right. So another key thing about TB is that, very interesting, it actually was the leading cause of death from a single infectious agent. And then HIV came into the picture. So HIV only started in the 1980s, and it's still around until today, although it's now more controlled. Now, HIV became the leading cause of death, but now TB is again the cause of death, but for people who are infected with HIV. And this is because, if you recall, HIV impairs the immune system. So patients with HIV are more likely to develop active TB. And the risk of active TB actually increases 2 to 5 times fold higher if a person's infected with HIV. And the fact that they're co-infected actually makes both diseases worse for the person. So TB also then increases your likelihood of getting other infections. HIV also the same. It will increase the risk of you getting other infections. And also, if you recall, TB infects macrophages, which requires CD4 T cells to help them out to intracellularly clear the bacteria. And the macrophages can be infected by HIV, but HIV definitely infects CD4 T cells. That's basically how the HIV is able to compromise the immune system. We'll talk more about HIV in the next lecture. But basically, this rapid loss of memory helper T cells and the ability to replace them is what causes this combination of TB and HIV infection to be so deadly. Another deadly immune response to bacterial infections is sepsis. So this is again another case where it's not the bacteria that kills the person, it's the immune response. So the pathogen will stimulate the immune response, but the immune response and overproduction of cytokines is what then leads to symptoms like bacterial septic shock and toxic shock syndrome. So that can be very rapidly fatal for people. Now, after bacteria, let's go into something even bigger, parasites. So parasites are a very broad range of infectious diseases, and what we usually talk about protozoans and helminths. So protozoans are unicellular eukaryotes, and helminths are basically parasitic worms. So they're multicellular. Protozoans are unicellular. So helminths can be quite big. You can even see them without using a microscope sometimes, and they can actually live outside of a host as well. They don't need to infect us. Sometimes we are an accidental intermediate host for them to complete their life cycle. So let's talk first about protozoa, and basically for protozoa, the humoral antibody is quite effective against the blood-borne stage of the protozoan life cycle. But once the protozoan has infected the host cells, then you need a cell-mediated response. Kind of similar to the bacterial infections. If it's extracellular, it's not inside a cell yet. The antibodies can do a good job getting rid of them. Once they enter a cell, you usually need cell-mediated immunity. So we're going to talk about malaria as our model protozoan parasitic disease, because it's really the number one cause of death from a parasite, and it's not so common in Malaysia. If you look at Sabah and Sarawak, they have more cases of malaria there. It's usually in areas that are more rural, and they'll be endemic. It means it's always there. People will be getting infected at a certain level, and unfortunately, with malaria, children will develop a very poor response to malaria, and so the mortality can reach as high as 50%. So 1 in every 2 kids that get malaria can possibly die from it. But people living in the areas that have malaria often have a lifelong low-level plasmodium infection that they're able to manage somehow. So how do plasmodium avoid the immune system? So the plasmodium can do a few things. So one, it actually has maturational changes. So as it's maturing, as it's developing, it changes its surface molecules. So again, our immune system is not very good at recognizing changing antigens. Yeah, so this what makes them easily missed. Then the intracellular phases of the life cycle. So once it enters another cell, then obviously it's harder to activate the immune system. In a sense, the parasite uses the organism's cells to shield it from attack. And if you look at basically this picture here, you have basically a very malaria, Plasmodium falciparum, for example, caused by Plasmodium falciparum, for example, begins its life cycle in the mosquito gut, and then when somebody gets bitten by a mosquito, then the sporozoites enter, and then it goes to the liver, and then it changes. Can you see how different the shape is? So now it's turned into merozoites, and then those will then infect the red blood cells. And then it changes again into gametocytes. And this is what will basically produce more offspring that the mosquito will then pick up and then spread to another person. So if you remember from my my previous lecture about vaccines, for malaria, the vaccine is to stop this part. So it's not about protecting this individual, it's protecting the next person. So it's slightly different form of vaccine. But what you should notice is that it's not just subtle changes, like some very significant morphological changes that happen, and it's very difficult for the immune system to deal with this. And the most accessible stage, the sporozoites, which is this stage here, that is only in the blood for about 30 minutes before it goes to the liver. So it's really hard for the immune system to be able to effectively mount a response against that, the plasmodium. And then it can do things like shutting off the surface antigens, and then then the antibodies can't bind them anymore. Now, another parasitic infection, which is the helminths, the parasitic worms. They're basically categorized as roundworms, tapeworms, or flukes. So that's the nematodes, the cystodes, and the trematodes. So the adults are large, usually you can see them, but they'll have microscopic eggs often. And so although the helminths are extracellular, they usually don't multiply in the host. So like I said, often we're just a part of their life cycle passage. So because they don't multiply, within the host. There are not that many of them. So they won't stimulate that much immunity against them. And usually they'll stimulate a Th2 immune response, which is why IgE is a very common antibody type that's produced in response to these Helminth infections. So our model Helminth is going to be Schistosomiasis. So it's caused by a schistosoma worm, and it's usually through free-swimming larvae. So most of the symptoms are initiated by the eggs, which invade the tissues and causes hemorrhage. So the unexcreted eggs will induce a cell-mediated delayed type hypersensitivity reaction, and it also produces granulomas, just like in TB. And so these granulomas then obstruct the blood flow to the liver and the bladder. And the worms can live up to 20 years inside the host. So a very long time, people are sick because of this. And it's because they're able to shield themselves from immune response, so they never get really cleared once they infect. And how do they do that? A few things, like they actually decrease the antigens that they express on their outer membrane. They also cover themselves with glycolipid and glycoproteins from the host. So they use host antigens, surround themselves with it. For example, your blood group antigens, the ABO, and histocompatibility antigens. And then they pretend, they're part of you, so your immune system won't recognize or attack it. Very clever. So schistosoma is one of those things you get if you go swimming in certain, I think it's freshwater areas. Because what happens is the cercaria from the schistosoma, which is released by their intermediate host, the snail, then goes into the water and penetrates the skin of an unsuspecting swimmer. And then it will start going through its life cycle, becomes a schistosomulae, migrate to the portal blood in the liver, mature into adults. So you've got the male and the female and the male. And then finally, after a long time, it'll migrate, lay the eggs, and then the eggs will be excreted in the urine and the feces. And if that somehow gets into another body of water, then the eggs will hatch, produce the myricidia, which then infects the snails, and then sporocysts in the snails will develop before again you release another cercaria that is ready to infect another human host. So very gruesome indeed. So the immune response against Schistosoma mansoni in particular is dominated by the Th2 response. So what's interesting is what they found is that the Th1 response, which means you produce interferons and there's macrophage activation, might actually be more protective. But the Schistosoma, first, it tries to avoid stimulating the immune response, and when it does, it actually induces the Th2 response, which is less effective in getting rid of them. So it's very interesting. So this is some examples or a schematic of the immune response against Schistosoma mansoni. But a key feature of this is you'll see the production of the IgE, which is usually what causes eosinophilia for these parasitic infections. Okay, we're getting into the last one. So fungal infections. So fungal infections are quite important, but we often say they're not so common and they're not so problematic in normal healthy individuals. And usually just your barriers of innate immunity can control most fungal infections. But what happens is if you have an immunocompromised state, that's when the fungal infections become opportunistic, and then they cause a lot of problems. You can have, for example, in your just your commensal fungi, like Candida albicans, right? If you're taking antibiotics for a long time, you start destroying your bacteria, which means your Candida albicans now have no other bacteria to compete against for its nutrients, which allows it to grow very rapidly. And this overgrowth then becomes an opportunistic infection. But often the innate immunity can deal with this. So, for example, there will be recognition of the fungal cell wall pathogen-associated molecular patterns, the PAMPs, and then this will activate the complement system and also induce phagocytosis by neutrophils, and then the fungal cells will be destroyed. And because the neutrophils play a key role with fungal infections, people with neutropenia, which means they don't have a lot of neutrophils or lower than normal, they are more likely to get fungal disease. On top of innate immune responses, you also have adaptive immune responses like Th1 responses, and they produce interferon gamma, which is important for macrophage activation, and also gives you protection against fungi. And that's why people with HIV/AIDS or people using drugs that suppress their immune system are more likely to get fungal infections. So this is a nice schematic showing the relationship, quite complex. You mainly will have the innate immune system protecting you against fungal infections, but it does also stimulate your Th1 and Th2 responses and also antibody responses. But what they found is that the B cells or the antibody response plays very little role in protecting you against fungal infection. In particular, when they looked at AIDS patients, even if in AIDS patients and in experimental animals, if the animal doesn't have B cells, they're not any more susceptible to fungal disease, meaning it's not so important to have B cells to prevent the fungal disease. So it's mostly cell-mediated. And there are two types of cell-mediated responses here, and very similar to our parasite or our Helminth infection just now. The Th1 response is what's going to protect you, and it's usually, if you're able to induce a Th1 response, you'll get a mild, largely asymptomatic infection. But if you induce a Th2 response, then you have more susceptibility to mycosis, which is an actual disease from the infection. And Th2 responses are actually associated with severe and relapsing disease from fungal infections. That's it. I didn't have a recap for you. Apologies. Basically, we talked about viral infections. We covered influenza and also a little bit about COVID-19. And very important about viruses is their ability to mutate. And that obviously gives some challenges to our immune system. And then we talked about bacterial infections, talked about my favorite bacteria, which is Mycobacterium tuberculosis, and the fact that they're intracellular, and also the fact that they produce a granuloma, and how important it is to maintain a CD4 T cell for that structure to be maintained. But over time, in some people, that structure still falls apart, and then that's when you have an active infection for TB. And that's why also TB, HIV co-infections are so terrible. And then we talked about our parasites, the protozoa, the Malaria, and the Helminth, the Schistosoma. So both of them are a bit trickier for the immune system to develop because one, they're either not exposed to the immune system for very long, for example, the Malaria plasmodium. And then also they devise very sneaky things like, covering themselves with host antigens, or they purposely stimulate a Th2 response, which is less effective. And finally, we talked about the fungal infections just now, largely controlled by the innate immunity, and failing that, T-cell immunity, B-cell, or the B-cell humoral responses appear less important for fungal infections. Some of our references here, have a look. And you can look out for a quiz that will cover some of the key points that we covered in the last two lectures. All right, so have a good week, and thank you.